Okay, well today's presentation is going to be about your ever-changing brain. And what do I mean by that? Well, up until quite recently, we used to think that once you reached adulthood, your brain health was kind of downhill from there. You started becoming slower and less good at remembering things, and just generally it was depressing news. So today's talk, I hope, is going to argue a little bit against that and give us all a little bit more optimism about our brains and the effect that our behavior can have on them. So I'm going to start my story with talking about rodents, mice and rats. And the reason for that is because that's where we used to get a lot of knowledge about science and anatomy. So back in the 1940s, a very famous psychologist, a Canadian psychologist called Dr. Donald Hebb, did an experiment. He had little kids at home and he had lots of rats in his lab in Montreal. And what he wanted to do is take some of the baby rats home to play with his kids, and it was really just for fun. And so he took some of the baby rats home, and they had a great time hanging out with his children, playing, doing probably slightly more interesting things than the lab rats would do if they were staying at the lab. And after a few months, he took the labs from home and he took them back to the lab and started running them through his psychological experiments with rats. So things like learning their way around a maze or learning to solve little problems so that they would get a nut or a piece of candy or something like that. And he made an observation. He observed that those rats who he'd taken home to play with his kids as pets did much, much better at the tasks. They learned them quicker, they got more of the little rewards. They were just smarter rats than the ones that had the regular development in the lab. And so he thought that there might be something important about environment where we grow up. And it kind of makes sense, right? So he made a theory. His theory was that the environment that you grow up in as a small child or baby rat is very important to your cognition, your intelligence, and your ability to reason and be smart later in life. That idea kind of sat in the field for a couple of decades until some neuroscientists at UC Berkeley, led by Dr. Rosenzweig, did some experiments. And they did experiments with mice and rats, and based on Dr. Hebb's theory about how you grow up and how that affects your brain. So they divided baby mice and baby rats up into two different groups, some who grew up in the standard lab environment where they had everything that they needed. They had shelter, they had warmth, they had food, water, they even had a friend to play with, versus another group who got the party cage. They got friends through the roof, they got social activity, they had more interesting food, they had plenty of water as the other ones did, and they had hamster wheels and balls and toys and other things that rats like to play with. Unfortunately, they then sacrificed these happy rats and the less happy ones too, and they looked at their brains. And what they learned is that the brains of the mice and the rats who'd grown up in the more exciting environment were very different, both on a sort of macro level, the weight of the brain and the size of the brain was different, but also even on the cellular level. So the cells of the mice brains that had grown up in the more interesting environment had more branches and more connections with other cells than did the same region cells in the rats who'd grown up in the less exciting environment. So that tells us something about early development and sort of childhood. But what was really exciting about Dr. Rosenzweig's experiments is he then repeated the study with adult rats and with older adult rats. So he took rats who'd grown up and had very, very similar development, and then he split them into two groups. And even if they were adults or older adult rats, he found very, very similar results to what he found with the baby rats. Exciting environment, better brains. So this was really dramatic. This was the first time that we learned that you weren't just born with a brain and then it developed normally and then it was sort of downhill from there. But actually the kind of environment that you have, even if it's only a change that's made once you're in adulthood or older adulthood, affects your brain health. So rats and mice are really interesting. We learn a lot from them. But what about people? How does this differentiate in humans? So we were really limited in the studies that we could do in the brain in adult humans or in any kind of humans until the development of really good neuroimaging techniques. So we have one of our neuroimaging technologists in the audience. She does PET scans all day, and people like her make these studies happen. So I'll just stand still. So with the advent of MRI techniques, we were able to suddenly start looking into the brains of healthy, normal people without hurting them, without damaging them, certainly without killing them and measuring their brain that way. And this led to a whole slew of really exciting studies of how the environment and skills and behaviors change our brains. And one of the first studies was in the 1990s in London. And London it's my hometown. It's a very, very complicated city. It's been growing up on top of itself since it was a marsh in the medieval ages, and now it's a very, very complex set of streets and connections, and it's got modern bits and certainly less modern bits. It's certainly not a grid system like Manhattan is, for example. This is a map of central London, and you can just see from that how incredibly complex it is. So London's iconic black cabs are a great job to have as a driver because you make a huge amount of money. If anyone's ever been to London and set foot in one of these black cabs, it's ridiculously expensive. And that's in part because the cab drivers are very, very skilled. They spent a couple of years learning all of the streets of London in very, very close detail. So sometimes you see people driving around London on little scooters with maps in front of them, and they're trainee cab drivers. And they have to pass a very difficult exam in order to get their license. The exam is called the knowledge. And in order to pass the knowledge, you have to go through a series of questions where you're given 2 points in London, just 2 street addresses. And this is a city of about 15 million people now. It's a huge sprawl. So any 2 cities, any 2 points in any 2 neighborhoods in London, and they have to come up with an efficient and effective way of getting from one to the other. And they have to come up with every turn and every stoplight and every red light that they would have to go along the way. So they become real experts in navigation through that training process. And Eleanor McGuire, who was a neuroscientist working at the University College London, was interested in the brains of cab drivers compared to regular non-cab drivers. And she had a theory that the part of the brain that's responsible not only for navigation but also for space memory, the hippocampus, would be different in experts, so cab drivers, and the rest of us. And she used MRI to look at this. So she initially just divided up a bunch of cab drivers and a bunch of similar people who weren't cab drivers, and she did a comparison. And indeed, she found that the hippocampus was different, and it was larger and seemed to be more effective in the cab drivers compared to the regular people. And then she found an extra layer of interesting findings on that, where she found that the size of that part of the hippocampus actually seemed to be getting bigger the longer somebody was a cab driver. So if you think about it, once you get your license, you're still driving around, you're learning new things every day, you keep learning. And this part of the brain, which is really important for memory and is actually the first part of the brain to be affected by Alzheimer's disease, is growing. Whereas in most people, it shrinks a little bit with age. So that was really exciting. Suddenly we knew that the hippocampus, this memory area, this important area in aging and Alzheimer's disease, could actually change in response to experience. So that was one study looking at one part of the brain. But these days, a couple of decades later, we tend to look more at networks of the brain. So yes, the hippocampus is very important for memory, but in part of that importance is how it's connected to a bunch of different regions. And using MR imaging and other imaging techniques, we can look inside the brain, we can look at specific areas, and we can also look at how those areas are talking to each other through the white matter, the long strings that attach one piece of the brain to the other. So these pictures, this is a phrenology bust. So this was a very ancient technique where people used to measure bumps on someone's head and correlate bumps in certain regions with personalities. That's not what we're doing anymore. These days we're looking at areas, discrete areas, but we're also using connections. So the white matter is in this picture. That's looking down on top of the head and looking at all of the different white matter regions. And the picture on the far end of the slide there shows functional activity. So if somebody's lying in the scanner and they're actually doing a task, we can look at all of the different parts of the brain that are especially active during that task, and then we can use special mathematical techniques to see how well those regions are talking to each other. And I'll come back to that later. So I want to turn my attention now to a study that we did here. And we did a study of experts, just like the cab drivers, but our experts were a little bit different. we wanted to also look at experts who had special skills that were related to that same region, the hippocampus and an area next to it called the entorhinal cortex. And again, we here at the Louis Center for Brain Health are really interested in those regions because that's where Alzheimer's starts. For some reason, the proteins that cause Alzheimer's know to start in those particular parts of the brain. So the theory is if we can do something that might change that region of the brain, then we might be onto something for a benefit to our Alzheimer's patients later on. So we took the basics of neuroscience. So if we look at a brain, we know that certain regions are the first kind of landing points in the brain of sensory information. So when you look out at the world, you have visual information coming in. That's a lot of sensory information. And the first place that really lands in your brain is right at the back. And in this picture, that's green, and it's called the visual cortex. But the area that we were interested in is a different area of sensory cortex called the olfactory cortex, which is in yellow here. And it's got a dotted line around it because it's deep down within the brain as opposed to on the surface like the visual cortex. So in this slide, you can kind of see the inside workings of the brain and how the primary olfactory cortex is right next to the hippocampus and the related areas, and they're connected through this entorhinal cortex. So we were interested in people who might have expertise that could particularly hone in on that region of the brain. And this is Las Vegas. It's a big resort city. We have some of the best food and beverage opportunities in the country here. And we also have access to a very special group of people, and they're called master sommeliers. So sommeliers are wine experts. If you go to a fancy restaurant, there's often a sommelier there who'll come and they'll help you navigate the wine menu and decide on something that's going to pair well with your meal. The master sommeliers are the elite there. There's about 220 of them in the world, and they've passed a series of exceedingly difficult exams. And I'm just going to show you now, if it works, a brief trailer. It takes about 3 minutes of a movie that was made about sommeliers. And the reason for doing this is just because it's going to show you in 3 minutes exactly what master sommeliers do and how they get there, and it's going to do it much better than I would be able to explain it. And it gives me a little break too. So let me get this up. Oops. Okay, it should start in a second. These things always work when the IT guy is standing right next to you, and then he leaves. I think it's just loading. There we go.
You have this plant, for thousands and thousands of years. I mean, people have been dedicating their life to make something amazing out of it.
It is living art, and you can't appreciate it until you consume it.
It's a lot more than just grape juice. It's a drug, really. Sommeliers are professionals that are supposedly experts about knowing about beverage and wine.
Soms now have become the new rock stars in our industry.
Some of the most dedicated, obsessed people.
So there's this diploma called the Master Sommelier Diploma.
It's the highest achievement you can make in the world of wine.
Over 40 years, there's only been like 170 in the world.
I cried when my parents died. I cried when my children were born. The only other time outside of that I cried was when I passed this exam.
I've got to know every single one on the planet.
It's pretty amazing the amount of information you have to know.
The regions, the sub-regions, the districts, the villages. It's in 5 languages.
Why certain years were good, how to serve it, what to eat with every different kind of wine.
Nothing else in my life has been this difficult.
Well, nobody ever said that anything that you worked hard for was going to be easy.
When someone tells you this is something that a lot of people can't do, people either go, wow, that's impressive, or they say, wow, I want to do that.
Perhaps to the detriment of your marriage or your relationship.
You have to be sickly gifted profits.
Egomaniacs.
Completely obsessed.
If you're really serious, if you really want to become an MS, you can't do it alone.
They're like guys in the locker room, with wine bottles.
So why don't you tell me how to taste them? I mean,
Do
sure that as the week goes on, it'll probably just get worse.
Is this happening right now?
I don't know, you tell me.
Oh, falling apart here. Absolutely brutal. This sucks.
I told myself I can be him, I have to be him, I will be him.
Okay. So I hope that you got a little bit from that trailer about Master Sommeliers. They pass a 4-step exam process that takes many years to get through. And one of the things that they have to do is taste, and I think it's within 20 minutes they have to taste 6 wines. And for each one they have to say where the wine was grown, down to the village. Oh, apparently we're being treated to more. Let's just get rid of that. So they have to tell a lot of details about the wine, and of course they have to identify the wine. So they have to come up with, this was a 2008 Bordeaux red, like down, I can't do it, but down to the detail. And to do that, they have to spend years tasting and learning about the wines. And of course, a lot of that is smell, but a lot of that is also memory and other associations that they make with the wine. They remember details about the labels, they remember the geography, they spend years studying maps. So this all sort of comes together on smell and memory. So we thought that these people could be really interesting to look at in terms of how different their brains are from regular people. The way we did that is by using FMRI. So we had to design a couple of tasks to look at that. We wanted to compare sommeliers and regular people, but we wanted to do it in a way that the tasks wouldn't be so easy for the sommeliers that they can do it, and the regular people start smelling wines and things and just say, well, I don't know. I mean, if you put a smell in my nose and ask me which region of France it was from, it's like, you could be speaking some alien language, I don't know. So we needed to create tasks that were similar enough to each other that we could compare them, but also easy enough so that regular people could do them as well as sommeliers. And we needed to create stimuli to put these smells into people's noses. We needed special equipment, and we needed to just scan people and then analyze the data. So we worked with a master sommelier. His name is Jay James. He used to be here in town. He was actually one of the earliest directors of Bellagio, which has one of the largest wine programs in the country, director of the Bellagio wine program. And then he worked at Southern Wine and Spirits, who are affiliated with us through Larry Ruvo, who's Lou Ruvo's son. And now he works in Napa Valley at a vineyard called Chapelet. He helped us to select wines that we could use that would be kind of typical, that wouldn't throw the sommeliers off. And then we compared the smell of the wines and decisions that people were making about wines in the scanner with what we called non-wine. So we created wine-like substances through a long process of making recipes where we use something called verjus, which is basically grape juice used as the basis for making wine, combined with a little bit of alcohol, so different kinds of vodka, some different fruit essences, because when you smell wine, it's very complex. It may just be made out of grapes, but the aging process and the development of the wine has led to having different smells. So we used some apricot essence, some fresh lemon, some pear essence, and in some cases some cognac, which has a kind of oaky smell. So we created things that your average Joe might confuse for a wine, and then the sommeliers would need to think about whether or not it was a wine. We wanted to make sure that any changes that we saw in our sommeliers' brains were specific to the smell system and not general to sensory abilities, because we don't expect them to be just experts in sensory abilities. We expect them to be experts in smell. So we created a similar task that we could compare it to that was a judgment of fingerprints or zebras. So again, both similar stimuli in the same way that the wine and the non-wine are, but different in some way. So we compared that down the line. This is just one example. And what do you think this is, a fingerprint or a zebra? Fingerprint, yeah. You might have had to think about it for a second or two, but everyone pretty much gets there in the end with that one. We used a special piece of equipment. This is Johannes Fresnelli, who's a professor up in Montreal, and he lent us this machine, which is called an olfactometer, which was used to push air into different bottles, which had different kinds of wine or the non-wine systems in, and that bubbled smells up into the person who was lying in the scanner. So this is just a little diagram showing you the whole set of equipment that we had. We used medical grade air, which pushed into this olfactometer, which was controlled by a computer. It told it which of 8 little tubes to open up, and then whichever tube opened up pushed air into a specific bottle of smell, which pushed the smell into a face mask that the poor person lying in the scanner was wearing. So it was quite an elaborate setup, and not particularly comfortable. It's certainly not the way most sommeliers would work on a day-to-day basis. We had them run through a computer program both before they went into the scanner to get used to it, and then once they were in the scanner, so that everyone breathed at the same rate, and no one was snorting the wine in there, and then other people were smelling it gently. We got everyone trained up so that everyone was doing kind of the same thing in the scanner. We ended up with 12 sommeliers and an age-matched group of 11 controls. So they were just regular people. A lot of them were recruited through UNLV, so they'd be smart people, similar in many ways to the sommeliers, but obviously not experts in wine. We created a wine quiz so that we could learn a bit more about the controls knowledge of wine. And we also did a scratch and sniff test. It's a scientific scratch and sniff test called the University of Pennsylvania Smell Identification Test. And we found, obviously, that the sommeliers were better at identifying these smells than controls. So this is a couple of questions for you to answer, to think about your wine knowledge. These are actual examples from the wine quiz that we used. So champagne is to France as what is to Spain? Anyone? Cava. You get the point. And then this is another one. This is, I think, a harder one. Carmenère is the best-known grape of Burgundy, Champagne, Chile, or Bulgaria. I didn't know that one either. The answer apparently is Chile. We had a graduate student from UNLV, very smart woman named Megan Pierce, who developed this. She also likes her wine. We wanted to find out whether or not the sommeliers and controls differed on the different tasks that we gave them in the scanner. And in fact, they didn't, and that's good, because that's what we designed them so that there wouldn't be too much difference, so that we were measuring similar kinds of brain activity in each group. So to go back to neuroanatomy a little bit, this is the kind of circuitry that we were interested in looking at. So we were interested in looking at the entorhinal cortex and the hippocampus, which are very, very closely related to where the smell comes into the brain. So if you think about a nose here, and your eye is sort of here, and this is your brain, and this is all of the circuitry that we were kind of interested in looking at. It's just like the smell system in the brain. So again, when we were interested in the specific regions, but then we were also interested in the network more generally. So the first question that we asked was very similar to that taxi driver study. We wanted to know if sommeliers' brains were structurally different. Was there something different about the shape of sommeliers' brains compared with controls? And happily, we did find that the right entorhinal cortex is larger, thicker in sommeliers than it is in controls. It's interesting that it's just the right side, because the different hemispheres, the two sides of our brains, are specialized in different functions. So the left side of your brain is very important for language, and the right side of your brain is very important for other things, including smell. So we seem to be finding a very specific region that was enhanced in the sommeliers, and it really made sense. Another region that was different is an area called the insula. So the insula is a part, again, quite deep down in the brain. It's a very kind of mysterious region. People didn't know what it did for a long time, and it seems to have a lot of tasks. It really integrates information from the outside world, information that comes in through our senses, whether that be our vision or our smell or our taste, and feelings from our inside world. So whether or not you're full, the insula is very important for that. If a certain smell disgusts you, the insula will go crazy if you're presented with a disgusting smell or even a fearful face. So things that are really important to connect our inside feelings with the outside world seem to kind of land on the insula and mesh there. And the insula was also thicker in the sommeliers. And if you think back to that clip that we watched a few minutes ago, they're integrating a lot of information. In a few minutes, they have to integrate a lot of information about their personal memories, their personal feelings, smells, their memory of how a wine label looked, what a map on the wall looked like. So it kind of makes sense that the insula would be different for them. Similar to the taxi drivers, again, we wanted to know if the sommeliers' brains changed over time. And although we weren't able to look at that because we didn't do a longitudinal study where we looked at the same people over time, we did correlate the size of those brain regions with the number of years since they passed the exam. And what we found is that our older sommeliers, who are up here, they're sort of in their 50s and 60s, even had a larger entorhinal cortex than the younger people. And this to me suggests that the younger ones are going to grow as they get older. So most of us decline on this measure as we get older, but there's something about what these people are doing every day, that they're training in their jobs, that is actually expanding this region of the brain. And that's a really exciting finding for me for what we do here more generally. The next question that we wanted to answer is, are sommeliers' brains functionally different? Do they differ in the way they work? And indeed, we found certain regions were more kind of lit up or more active during the smell tasks, specifically in the sommeliers compared with the controls. And those regions were the same regions that are thicker, so that kind of makes sense. It seems that those regions are sort of practicing what they're doing all the time. So we have the smell regions. We have the insula that we just spoke about. We have the memory regions, more on the right than the left. And we also had something that was a little bit surprising, which was the visual regions. The area of the brain that's important for processing what we see were more active during the olfactory task, during the smell task in the sommeliers. And I was kind of perplexed by this. Why would that be? That just seems strange. So I had a conversation with Jay James, the sommelier, and I was like, Jay, our findings are really cool. We found a lot of stuff that really makes sense. I'm really excited about this study. But there's one thing that I just can't make sense of, which is this visual area. And he didn't even think about it. He was just like, well, Sarah, think about what I told you about the way we train people. One of the things that they do when they're training people to become sommeliers is to walk through the produce section of like a Whole Foods or some nice supermarket where they have really good fruit and vegetables. And they have them go through and sniff all the different fruits, and these people must look very strange when they're doing this, but really learn about different smells and connecting that with the sight of the smells. And then when they're doing their blind tasting, when they're trying to figure out what smells they're smelling in their exam, a lot of them will visualize and mentally walk through that grocery section until they find in their head the orange or the apricot or whatever fruit it is that they think that they're smelling. So what the sommelier suggested to me is that that's what people are doing in the scanner. We're presenting them with these really weird smells, and they're trying to work out, their brains are trying to work out kind of automatically what's going on. So that's an explanation that maybe makes sense. We've taken it one step further now. So we have some new staff here from Dietmar Cordes, who came to us from Canada, and then he's hired Xiaowei Zhuang and Karthik Srinivasan, who've both been working on this data with me. And they're doing really exciting high-level imaging analysis. And one thing that they've been doing is looking at how the different parts of the brain will talk to each other during this task. And they've been isolating brain networks that are particularly active in the sommeliers compared to the controls. And indeed, they found these complex regions, many of them kind of hubbing or centralizing in the insula, that are more developed in the sommeliers. And I'm going to show you two more slides now, and I don't want you to think about too much of the detail. It's really just to look at the complexity. So this is just called a circle diagram, and I want you to compare the orange lines compared with the blue ones. So the orange lines represent regions of the brain that are more connected in sommeliers, and the blue lines, of which there are 2, 3, represent the areas of the brain that are more connected in the regular people. And this is specifically when they're doing the smell task. So you can see that the level of intercommunication in the brain for the sommeliers is huge compared with the controls. And this is just one more depiction of that same kind of result. And this is the right side of the brain. And you can see that the right side of the brain is the kind of the hub of connections. And that's where the information is coming in and initially being remembered. Associations are being made with different senses. It's connecting to things like the language areas of the brain on the left, where presumably sommeliers are attaching labels to the smells. And it's just incredible to me how the sommeliers are just so much more active than the controls during these tasks. And it's not just the smell regions, it's really the whole brain. And this is a little video which I didn't examine. Let's see if it works. It's just that the same finding but being rotated so you can really get an idea of all of the different connections. So we're still doing these results, we're still analyzing these data and trying to refine exactly what it means. So this is the study that we did, and it was very interesting to me. It was a fun study to do. We found very, very cool results, and it's interesting just from a scientific perspective to know that the brain seems to change that way, and that in these particular experts it seems to change in really dramatic ways, both on a regional structural level but also on the network level as a whole. But that's not really what we do here at the Lou Rouveau Center for Brain Health. So why does it matter? Why do these results matter? Well, I think one of the big take-homes from my message should be that brain plasticity, the ability of the brain to mold and change, happens way into adulthood. This is true even in those regions which are really vulnerable to diseases like Alzheimer's disease. So presumably these guys didn't start learning about wine when they were 5. They started when they were in their late teens, maybe, more likely early 20s or mid-20s or even later. So that's really interesting to me. We don't know that they won't get Alzheimer's disease, and I would love to do that study in 30, 40, 50 years' time if I'm still alive and they are, but there's something about those same regions of their brain that seem to be better. Something that I found in interacting with these guys is that they're very passionate about what they do. They really love it. And I think you probably got a little bit of that from the clip too, the level of commitment that they put into getting through these exams. And I think that that's something that we find in a lot of studies of healthy aging, is that people who age well and who are somehow resistant to the horrible diseases of aging tend to be very optimistic. They often do something that they really love and enjoy working at things late into adulthood. So I think that that might be relevant. Another thing that these guys do is they're very social. Their jobs tend to be in restaurants. It's their job to interact with different people of all walks of life every day. So that's something that we tell people is very important. And again, there's some really interesting research coming out that loneliness is a big risk factor for Alzheimer's disease. And having even just one close friend or ally or a spouse who you have a lot of fun with is a protective element there. So I think that these are all things that are extracted from the study that are important to remember. Finally, we are actually applying some of what we learned in the plasticity to cognitive intervention. So we have a new study in Parkinson's disease that will be starting in the next few months looking at brain training. So you see a lot of these brain training programs, and we're actually going to focus in on some of the problems that people have with thinking when they have Parkinson's disease and see if we can target those areas, the areas of the brain that are responsible for that dysfunction, and train that. We're going to do imaging beforehand and imaging afterwards and see if we can create some brain plasticity of ourselves. And that might just be the first step. So something that we might do in the future is look at sensory training and combining that with cognition. So if we trained a group of older adults to do something akin to not becoming a sommelier, but combining smell and other memories and things like that, maybe that would have a positive impact. So those are some of the directions that we're hoping to potentially take this study in the future. This was a small study, but it was also a big study. It involved a lot of different people. So along the whole course of the study was Dr. Leger, but we had students from UNLV, and my team here were very central. So I won't go through everyone and thank everyone individually, but there were a lot of people who made this study happen, even down to their finances and giving us wine. So thank you to all those people. I was pregnant during the study, which was bad timing because there were lots of leftovers. So thank you. And at this stage, if anybody has any questions, either here or at one of our other Nevada sites, I'd be happy to take them. Hi.
All right. How well does this plasticity carry over from the sommelier into their regular life? Does it help them?
Well, I think that's a really interesting question, and something that we didn't do but would have been interesting to do. Oh, I'm sorry, I have to repeat the question. So the question was, how does the plasticity, or how do our findings carry over from the sommelier in their kind of professional life into their regular life? Is that right?
Right.
So we didn't test their memory more generally, but I think that would have been a really interesting thing to do, to see if the superiority that they show is just for wine-specific things, or if they're better at, say, navigation or other kinds of memory. The interesting thing that we would love to look at if we ever got the chance would be to see what this means for their future. So to see if there's a sort of protective effect of the changes or the differences that we saw. We can't tell with our data right now, but if we were able to follow up with them in 10, 20, 30 years and see, especially some of the older guys, see how well they fared, it would be really interesting to see if this is actually protective against disease, or if they age better because of these differences that they show. Hi. Yeah.
Seems like it's a happy thing that they're doing, and there's really nothing bad that will happen if they screw up. No, they're not like air traffic controller, a doctor, an accountant who's
stressful.
Are investing in this based on their analysis. So it seems easier. It seems, they're not under stress. Yeah.
So just to repeat that comment, the person said that what the sommeliers do seems to be a kind of a safe job. There's very little stress involved. They're not going to hurt anyone, or nothing bad will happen if they screw up the wine. I guess they might upset a customer, but beyond that, yes, they live I think stress is relative. I think everyone has stresses in their jobs, whether it's an ER physician who's making life or death decisions versus somebody who's making decisions about this $40,000 bottle of wine versus this $3,000 bottle of wine. But yes, I think that there's obviously a different level, and there's probably different personalities who are attracted to those different jobs in the first place. But I think that what you bring up, the relevance of stress in the brain, is very important. Stress can be a good thing, but it can also be a bad thing. Yeah, too much. And the way we interpret it and react to it, I think, is very important as well. Any other questions or comments? Yes, please.
What would you have us take away for our personal lives? Like, what would I want to do to improve my brain?
Well, I would love to prescribe a bottle of wine, but I might lose my job if I did that. I think it's doing what you love and doing what you're interested in, and challenging yourself a little bit, and not worrying about or encouraging yourself or pushing yourself to try new things, even if they're difficult. There's a lot of research way beyond this study to suggest that can be protective. Learning a new language, learning a musical instrument, traveling, having political discussions in a safe environment with friends, or just doing things which are stimulating, and making sure that you don't get into too much of a drudgery routine. I think we all have things that we have to do that we don't really like doing, and I think making time for doing things which are going to be really rewarding and satisfying are going to be good for you in general, and hopefully for your brain as well. Yes. Any questions from anywhere else in Nevada? It seems like we're quite linked in. We have a network of connections throughout the state. Okay, well, let me know. Don't be worried to speak up. We have a couple more questions here, I think. Yes?
What you said about the environment, I was thinking about ethical questions like prisoners who are in solitary confinement. And at the other end of the age spectrum, there's baby nurseries in Israel where babies are they see nothing but an empty room and a crib. Nobody pays any attention to them all day. So what are the implications of that?
Well, I think that unfortunately there was a lot of interesting science which came out of a really horrible situation in Romania a couple of decades ago. People might remember there were a lot of orphanages, and my history and politics aren't good, but the end of Ceausescu's regime, is that right? They discovered these poor babies who were in this awful, neglectful conditions, and there were some research was done on their ability to kind of recover from those situations. And they did discover that kind of neglect early on, and it was building on some other studies done in the 60s and 70s, mostly with monkeys, it can have really detrimental effects. So I think there's normal, like I showed the picture of the normal mice and rats who had some level of stimulation, and then there's neglect. So I think having total isolation and total neglect, it really changes the way the brain works and the way the brain is wired. And it probably is something that, if it's bad enough, will have an impact that won't be reversible later on. But I'm hopeful for all those babies that there's some improvement. But of course, there's other things, there's other factors like nutritional, whether or not they got the right foods and all of those kind of things, which also play a role, and whether or not they were abandoned because they were sick as babies. So there's a lot of complex issues, but I think obviously stimulating and offering warmth and comfort to our children is very important, and the social involvement as well. Does that answer your question? Sort of. Prisoners, I'm not sure about. I'm assuming that solitary confinement would not be very good for your brain, though.
Somebody was on television talking about that.
This is Kelly and Hawthorne, and we have a question.
Okay, Hawthorne. Let me take the Hawthorne question then.
Okay.
Can you tell us where Hawthorne is?
If you consider cooking classes, like cooking with spices for olfactory stimulation.
I think that's a brilliant idea. I don't think that I have not considered it, but I think that would be a really interesting way of looking at it, which would be probably more ethically viable than having our patients learn about wine and drinking wine. So thank you. We wouldn't have to get them a special driver to take them home at the end of the session. Thank you. And I think there was one more question from Las Vegas as well.
I wanted to go back to your slide on page 5. When you showed the two regions of the brain, I wanted to make sure I understood you. Did you say that even the two regions of the brain, that it showed damages for something like Alzheimer's or the onset of it, as well as when you found increased activity, these are the two regions that showed an increase in the neurons or the development of other neurons?
Is this the slide that you're talking
Yes.
about? Okay, great. So I'll talk first about Alzheimer's disease. So Alzheimer's disease, the very earliest changes that we see are plaques and tangles primarily are developing in this region of the brain, the entorhinal and the hippocampus region. And yes, those are the same regions that we saw in the, especially the entorhinus, but the entorhinal cortex, but a little bit in the hippocampus as well, that were larger in the sommeliers, and they were also more active and more connected.
Did you find a correlation, not a correlation, did you find that when there is more activity, there was less plaque development or a stymie of the plaque development because of their...
Well, unfortunately, we don't have those data. So up until maybe 5 years ago, the only way to actually know that would have been to just kill the sommelier there and then and do an autopsy. But we didn't do that. Excitingly now, we do have a kind of scan that Elaine here does quite frequently called a Flor-Beta-Peer scan, which can actually look at those plaques. And we have another kind of scan or a medicine that we can put into the brain and then use PET scan to look at tau, which is the tangles. We haven't done that with this group. It's exceedingly expensive. So it would be interesting to look and see if they had less of the actual Alzheimer's disease in the brain. But we didn't get to do that this time.
As a follow-up question, could
Sure.
you point to an article or magazine where if someone does that, where they would publish that so that we could read it?
We're in the process of getting this published. In terms of the tangles and the plaques, I don't think anyone's done that yet. So I'm sure if they did, it would hit the popular press, but I don't think anyone even has plans to do that. It's interesting though. We should get the black cab drivers, or we should go to London, Elaine.
I think that sounds great.
Okay. Any other questions? Okay. Well, thank you so much for coming and for listening, and have a wonderful day.